Welcome to our lesson overview on 2.5.1 DNA structure, and we're actually going to get into a little bit of the history of DNA as well. There's quite a few names and so forth in here, but I won't have you remember dates necessarily, but certainly you should remember some names. This can be found in the textbook sections 12.1 and 12.2 as well if you want to take a look there. As scientists realized well before we found out about DNA that there was something that we called genes that helped make us who we were. And so how do we go about finding out what molecule or molecules go into making a gene? Well, lots of people thought it was protein. When we found out about DNA, some people thought it might be DNA. And so in the end, we proved that it was DNA using viruses. Viruses are when they reproduce, viruses have DNA in them, and they're very simplistic. They're just DNA in the head of this protein-coated shell, and they attach to different cells. So a man by the name of Hershey, and Alfred Hershey, and his partner Martha Chase decided to take a look at this process. And so they took a virus that they knew had DNA in it, and they switched the phosphorus in the DNA to a radioactive form of that phosphorus, so they could track the DNA as it went through this regeneration of bacteriophages in this bacterial cell. Then they took other bacteriophages, same type of bacteriophage, but instead of tagging the DNA, they tagged the protein code because they knew the protein had sulfur in it. And so they put radioactive sulfur in there. And so they took that radioactive sulfur bacteriophage, they put it on a bacterial cell, let it do a thing for a few minutes, and then they knocked the bacteriophage off before it could destroy the bacterial cell. And what they found was in that bacterial cell there was no radioactivity, which means that none of the protein got into the bacterial cell. So they looked at the bacteriophage that had the phosphorus, the radioactive phosphorus in the DNA. And so they let the phage do its thing again, infect the bacterium, and then they knocked the phage off. And what they found in the bacterial cell was this DNA that had radioactive phosphorus 32 coming from it, which meant that DNA was the chemical. DNA is the chemical version of a gene. They proved that DNA was our genetic material. So what is the role of DNA? Why did Hershey and Chase, why did all these people care so much about DNA? Well, DNA is a few different things for us. Number one, we're going to talk about DNA in terms of being like a cookbook. Each cell needs a cookbook so that it can figure out what it needs to make in order to survive, in order to do its job. So DNA, first and foremost, is a safe place to store information. And we're going to call this information recipes while we're studying this. And so our DNA is going to store information in this book. It's a safe place to have that information. Next, we are going to be able to copy that information. Nobody prints just one cookbook. The same cookbook, there's millions of prints of it, and so each cell has to be able to copy its information so that new cells or new organisms can have a copy of that information. And then finally we have to, once we make a copy, so here's an old cell, here we make the copy. Once we copy it, that's not enough. We've got to be able to transmit it into new cells. So we have to transmit the info. So its three jobs is to store, copy, and transmit information. Well, what are the chemical components that help us do that? Well, we have learned already in class that DNA and all nucleic acids are made up of nucleotides. Those nucleotides can be joined together in any order, meaning that any sequence of bases is possible. That's how our DNA is different from one person to another, from one organism to another. Each nucleotide has a phosphate group, a 5-carbon sugar, let's write that down, 5-carbon sugar, and in DNA that sugar is going to be deoxyribose. And then we are going to have one of 4 nitrogenous bases, either adenine, guanine, cytosine, or thymine. So this right here is one single nucleotide. The order of these bases is going to be the information in our cookbook. The order is important. These nucleotides are connected, here's a connection, by extremely strong covalent bonds. Alright? But what clues helped us figure out and solve the structure of DNA? Well, the first thing that helped us solve the structure of DNA was by a man by the name of Erwin Chargaff. Chargaff discovered by measuring the amounts of adenine, thymine, guanine, and cytosine that, and this became known as his rules, in all cells the DNA is going to be consistent. There's going to be the percentage of adenine in any cell is going to be equal to the percentage of thymine. The percentage of cytosine is going to be equal to the percentage of guanine. What that meant was, well, if I know my amount of adenine, if I know that I have 10% adenine, well, then I must have 10% thymine. Well, that gives me 20% of my nitrogenous bases. The other 80% must be cytosine and guanine. So that means if I just split these, because these are equal, if I just split them, 40% is going to be guanine, 40% is going to be cytosine. That would give me a total of 100%. That's Chargaff's rules. He knows that the A's and the T's are equal to one another, C's and the G's are equal to one another. The next person is Rosalind Franklin. Rosalind Franklin took these X-ray photographs of DNA, and we've talked about this before. This X means something. The X that we see in her photographs means that there were multiple, that our strands, or our strand, our molecule, was twisted. That X means that the molecule is twisted. The angle that we see here, the angle means a couple of things. Number one, the angle means that we have 2 strands. And it means that our nitrogenous bases are located in the middle of the structure. They're on the inside. So her X-ray photographs meant a lot, but she never lived to see it. And so Watson and Crick, James Watson and Francis Crick, took the information from Franklin, from Chargaff, and from others, and they came up with a very important, they didn't really do any tests themselves, they just looked at other people's research, and they made models. And the model that they developed for DNA, they developed what was known as the double helix model in 1953. The double helix model was important because it told us several things. Alright, they used Franklin's pictures to help them make that double helix model. And the double helix model would not only explain Franklin's photographs, but it also explained Chargaff's rules and how the strands were held together. The double helix, the name itself actually means twisted ladder. It means twisted ladder, and this twisted ladder accounted for the X-ray photographs made by Franklin, Rosalind Franklin. Accounted for the X-ray patterns from Franklin. Very important. The double helix model proved what they saw in those pictures. They also assumed that the strands went in opposite directions, that they went from this one strand would go in one direction, the other would go in the other direction, and what that helped with was, one, it lined our bases up. It lined the nitrogenous bases appropriately. It lined them up. The other thing that it did was it gave us part of a copying mechanism. So if I had one side, then I could easily make the other side. So it helped explain a copying mechanism, or a way to copy the information. The hydrogen bonds gave us a couple of things. These hydrogen bonds were strong enough to hold together the two sides of the molecule, but just strong enough to hold it together. But it was easy enough to break apart, or it could be separated in order to copy the material. Could separate to copy info. Hydrogen bonds were also a perfect fit in between our bases. There's 3 hydrogen bonds in between guanine and cytosine, 2 in between adenine and thymine. It was a perfect fit. Alright? And what that meant is it told us that A's would always bond with T's, always, always, always. A's bond with T's, C's bond with G's, and this information accounted for the experimental data collected by Chargaff. Chargaff's rules. Alright. I know that was a long one. There was a lot of information. We've got the history of DNA. We've got what makes up a nucleotide and how those nucleotides can be put together to make a single DNA molecule. Make sure that you go over this, and we will talk more about this in class in the upcoming days. We'll see you in class.